Namaskar dear students, I welcome you to our channel by Gulab Sir. As a part of series of lectures, today's topic is Electron Transport System. Very interesting to know about transfer of electron, but moreover, very interesting to know about what happened with oxygen which we inhale. No doubt in plants and animal both, that oxygen is required as a part of respiration to produce the energy which is required for all metabolic reactions on which we survive. Therefore oxygen is known as प्राण वायु. हमको क्या बताया जाता था या जब 6-7 में पढ़ाया जाता है तो ये बताया जाता है ऑक्सीजन हेल्प इन बर्निंग ऑफ फूड. सो व्हाट वी अंडरस्टैंड, हम ये समझते हैं कि व्हेन वी इनहेल ऑक्सीजन इट हेल्प इन बर्निंग ऑफ फूड, मतलब इनसाइड द स्टमक इट हेल्प इन बर्निंग ऑफ फूड. That was my understanding. It may be yours also in the 6-7. Then what happens with oxygen when we inhale? Where it goes? No doubt. As you learn or we learn that oxygen combined with hemoglobin form oxyhemoglobin through blood, it reaches to the cell. This is what we learn. It do not reach to the stomach. It reaches to the cell, each cell. But in each cell, where that oxygen goes? What is the destiny of oxygen? Oxygen get into the mitochondria. Let's understand what is the importance of oxygen, what we inhale. Through this process I am going to explain. Let us wait. Really what is the importance of oxygen you are going to learn today. What is the destiny of oxygen you are going to learn today. Why it is known as pranavayu you are going to learn it today. Let us continue. This is a topic, that electron transport system. What is the definition? Metabolic pathway. A metabolic pathway through which electron is transferred or carried from one carrier to another carrier. Finally that electron is used to ionize the oxygen which we inhale. That ionized oxygen combined with proton, that is what hydrogen ion, to form the metabolic water. The destiny of oxygen is to combine with hydrogen, control the proton. Otherwise, increase in concentration of hydrogen, what will happen? Decrease the pH. Decrease in pH, what is it? Acidity. Acidity is the main problem of our body. It is like a daitya. When we say oxygen is pranavayu, then hydrogen is daitya, pran lene wala. अगर हाइड्रोजन आयन कंट्रोल नहीं होंगे तो we feel suffocate because it decreases pH. Acidity kill the mitochondria, acidity kill the cell, and thereby within a very short period of time, if we do not inhale the oxygen, fresh oxygen to control the proton, we feel suffocated and we get die. And that is what the importance of oxygen. Now let us understand how it happens through process. This is the electron transport system. As I told you, metabolic pathway through which electrons are carried from one carrier to another carrier. Here there are many carriers. That is what complex 1, complex 2, complex 3, complex 4. These are the carrier to carry electron right from release from oxidation of NADH2 and FADH2 to ionized oxygen. So first step in electron transport system is oxidation of NADH2 to NAD. Where this NADH2 produced? Where had this FADH2 produced? As we already learned, this NADH2 and FADH2 are produced in glycolysis and Krebs cycle. There they are not oxidized, they oxidize in mitochondria. See the structure of mitochondria. This is the outer membrane of mitochondria. This is the inner membrane of mitochondria. And this is intermembrane space between mitochondria, between outer membrane and inner membrane. And this is what? This is a inner membrane. This is just I have been expanded to how that accommodation of this electron transport system content to understand. So this is the inner membrane through which electron transfer. So see, this is a mitochondrial structure and everything happening in mitochondria, including outer membrane, inner membrane and matrix. So what are these part? Outer membrane, inner membrane space, and this is a matrix. So you got it. Now, where the NADH2 and FADH2 are, they are present in matrix. Now they undergo oxidation in presence of enzymes. NADH dehydrogenase and FADH dehydrogenase, that is called succinic dehydrogenase. As a part of Krebs cycle, we already learned. Here NADH2 undergo oxidation, releasing proton. These protons are transferred on thymine mononucleotide, which is already ionized, from form thymine mononucleotide hydrogen. Further these get oxidized, release that proton outside the inner membrane, that is in inner membrane space, and release electron transfer to iron, Fes. So who is FES? Is electron carrier. So electron carry from iron to ubiquinone to naturally. Because of that reduce ubiquinone. Because of receiving electron, ubiquinone got reduced. Reduced one can oxidize. So this ubiquinone has 2 electron, can combine with 2 proton. So from the ubiquinone hydrogen, that is UQH2. Now this hydrogen released by the oxidation of this NADH2 to NAD, 2 proton combine with its ubiquinone to form UQH2 at the same time. When this complex system is going on, complex 2, that is called succinic dehydrogenase, also oxidize FADH2 to FAD, releasing 2 proton. That 2 proton combine with that reduced ubiquinone to form UQH2. So hydrogen released by the oxidation of NADH2 and FADH2 combine with ubiquinone to form UQH2, releasing that proton, that is hydrogen ion, from inside to outside, that is through this membrane. So this proton released from ubiquinone accumulated inside the inner membrane space. This is the intermembrane space. So proton keep on accumulating in the intermembrane space, so as to increase its potential, its concentration, to transfer from higher concentration to lower concentration, releasing that potential energy to generate the ATP. And that is the aim of this electron transfer system, to oxidize NADH2 and FADH2 to transfer that proton from matrix to inner membrane space for the generation of ATP. So this is a complex 2. Now what is the role of complex 3? Complex 3 lies between complex 1 and complex 2, receiving proton from complex 1 and complex 2 along with electron, so as to release this proton outside. When this UQH2 get oxidized, by releasing proton also release electron. So electron from complex 3, where it goes? Because there is no way from complex 3 to 2 or 2 to 3 to transfer the electron, but electron released after oxidation of ubiquinone H2, protons are released out. 4 electrons which release, they transfer on cytochrome C. Cytochrome C is a real complex 3. Now cytochrome C is a carrier, electron carrier. So what is cytochrome C? Is a small protein attached to the outer side of the inner membrane. See, I have shown here outer side of this inner membrane, and this cytochrome C is attached. This cytochrome C carry electron from complex 3 to complex pro, that is cytochrome C to cytochrome A. Now you got it, the role of this cytochrome C to cytochrome A, which attached to the outer side of the inner membrane space. Now when electron is transferred from cytochrome C to cytochrome A, it is transferred on copper, and copper to cytochrome A A3. What is this cytochrome A A3? Is a complex 4, where the electron is transferred. Now electron carry from cytochrome A to A3, A3 to copper, and then release into the matrix to ionize the oxygen, so as to combine with proton and form the water. So finally, when this proton H positive ion accumulated in the inner membrane space, intermembrane space, transfer from their higher concentration to lower concentration through complex 5. So what is complex 5? Complex 5 is a ATP synthetase, comprises 2 components, F0, F1. What is F0? Is electron carrier, proton carrier that carry proton from intermembrane space to matrix. And what is F1? Is a catalytical molecule to catalyze the process of synthesis of ATP. So ADP and inorganic phosphate combine with each other in presence of energy released when proton transfer from their higher concentration to lower concentration. Thus, release potential energy is used to synthesize ATP through complex 5. Finally, ATPs are also synthesized, and proton, which become free or transfer from intermembrane space to matrix, combine with oxygen to produce H2. So what is the role of this oxygen we inhale? It is to control the proton in mitochondria. What is mitochondria? Powerhouse of cell. If mitochondria die, mitochondria degenerated, what will happen? Our powerhouse will destroy. When powerhouse is destroyed, from where the energy will come? When energy will not be there, how we can be live? So who is enabling to live? Oxygen. How it is enabling to live? Transferring or getting into the mitochondria to control the proton. If protons are not controlled, then definitely this NADH2 will not be oxidized. If NADH2 is not oxidized, then NADH2 will not produce. If NADH2 will not produce, then means our food is not digested, means you will not feel hungry. Why we feel angry? Because we play. We have some exercise so that we inhale the more oxygen to control the more proton, to have the more oxidation of NADH2, to produce the more NADH2, to have the more food. Therefore we feel hungry. See the link. हमको भूख क्यों लगती है? भूख इसीलिए लगती है कि हमने ऑक्सीजन ज्यादा किया। जब हम खेलते कूदते बहुत एक्सरसाइज करते भूख लगती है। इसका मतलब हमने क्या लिया? इनहेल क्या किया? ऑक्सीजन। ऑक्सीजन ज्यादा लेने के बाद क्या हुआ? जितने भी NADH2 थे ऑक्सीडाइज हो गए, प्रोटॉन प्रोड्यूस हुआ, वाटर मेटाबॉलिक वाटर तैयार हुआ। इसका मतलब NADH2 की कमी आई। NADH2 की कमी आई इसका मतलब ग्लूकोज डाइजेस्ट हुआ। ग्लूकोज डाइजेस्ट हुआ मतलब भूख लगी। इसलिए ये एक रिलेशन है एक्सरसाइज का। अगर हम लोग एक्सरसाइज नहीं करेंगे, खाना खा के बैठ जाएंगे तो भूख नहीं लगेगी। क्यों? तो हमने इतना ऑक्सीजन इनहेल नहीं किया, इसलिए प्रोटॉन कंट्रोल नहीं हुआ। प्रोटॉन कंट्रोल नहीं हुआ इसलिए NADH2 FADH2 ऑक्सीडाइज नहीं हुए। NADH2 FADH2 जो पहले से प्रोड्यूस हुए वही यूज नहीं हुए, तो फिर जो ग्लूकोज है वो कैसे ऑक्सीडाइज होगा? इसलिए ग्लाइकोलाइसिस क्रेब्स साइकिल होगी नहीं, तो ग्लूकोज वैसा ही रहेगा और फिर हमको भूख नहीं लगेगी। सी द रिलेशन ऑफ एक्सरसाइज विथ आवर हंगर। So there is a link. This is a metabolic pathway to keep us healthy. We should know everything. If we know this physiology, I think so, you will be away from the doctor. So please let you know the importance of exercise and inhale oxygen to control the H positive ion so as to have fresh mitochondria, fresh rejuvenated cell to have the normal functioning. So higher concentration of H positive ion, that is decreased pH, we call acidity. Acidity simply does not mean that having खट्टा पानी, मुंह में खट्टा पानी आया, स्टमक से ऐसा लगता है कि खट्टा पानी आया तो अम्ल ज्यादा हुआ मतलब एसिड ज्यादा हुआ, या कुछ अलग अलग, ऐसा नहीं। Real acidity is increase in H positive ion concentration in your cell that can cause many deformities and disorders. Therefore have regular exercise to have more oxygen so as to have control the proton. And pranayam का महत्व क्यों बताया गया? What is pranayam really? When we have deep breathing, what is the importance of pranayam then? Why it is, मतलब ऐसा क्यों बताया गया कि प्राणायाम बहुत अच्छी चीज है? प्राणायाम थोड़ा धर्म के साथ जोड़ा गया। प्राणायाम के साथ जब ओम कहा जाता है कि मेडिटेट करो, मेडिटेट करने से ईश्वर के नजदीक जाते हैं। इसका अर्थ क्या है? ये सब का अर्थ है कि आपके अंदर ऊर्जा प्रोड्यूस करना। और ऊर्जा प्रोड्यूस क्यों हो रही है? तो फिर आप ज्यादा ऑक्सीजन इनहेल कर रहे हैं। ऑक्सीजन इनहेल करेंगे तो वाइब्रेशन आएंगे ही आएंगे। तो हमको क्या कहा जाता है कि हमने जब मेडिटेट किया और ओम बोला या ज्यादा ऑक्सीजन इनहेल किया या ज्यादा देर तक हमने इनहेल और एक्सेल ब्रीथिंग प्रोसेस की, तो क्या होगा? ज्यादा ज्यादा ऑक्सीजन शरीर में जाएगा, तो ज्यादा H पॉजिटिव आयन कंट्रोल हो गए, तो ज्यादा ATP प्रोड्यूस हो गए, तो ज्यादा एनर्जी हो गए, तो वाइब्रेशन तो आएंगे ना। इसीलिए तो कहा जाता है इनके सानिध्य में जाने के बाद जब हम मेडिटेट करते तो वाइब्रेशन आते और बहुत अच्छे वाइब थे। इसका अर्थ क्या हो गया? आपकी मशीनरी ही यूज की गई है वो वाइब्स आने के लिए। तो प्लीज लेट अस अंडरस्टैंड द फिजियोलॉजी ऑफ मेडिटेशन आल्सो, फिजियोलॉजी ऑफ प्राणायाम आल्सो। So not only a syllabus but also it relate to our life. Therefore it's very interesting. So just with this knowledge, just you compare this knowledge with that processes like meditation and pranayam or exercise or hungriness and thirst. So everything you will come to know and it will be clear. Let us come to the syllabus. So one second question can be asked, what is ETS? What it comprises of? ETS is a transport of electron from one carrier to another carrier during metabolic reactions, and that is what through these four complex which I have mentioned here. Go through this complex 1, 2, 3, 4 and everything I have mentioned very clearly as a part of notes. You please refer these notes so as to no need to worry about your preparation in examination. Complex 1, NADH2 dehydrogenase that help to oxidize NADH2 to release the proton H, which combine with ubiquinone to release it, flavin mononucleotide to release it into the internuclear space. Complex 1. Complex 2, that is what cytochrome BC1, where that ubiquinone from FADH2 and NADH2 come together to release the proton. That is what cytochrome BC1. Then complex 3. Complex 3. That what is to transfer this electron and proton from NADH2 and FADH2 through cytochrome C to cytochrome C1, that is complex 4. So complex 3 transfer electron from complex 2 to complex 4. So therefore it has in connection that as an electron carrier that Cytochrome C is a small protein attached to the outer membrane of the inner space to carry the electron. Finally, electron reaches to A3 and then transfer to the mitochondria through complex 4. So therefore it is mentioned here complex 4 also. The complex 4 is a complex 4. Here it is mentioned that what electron transfer from A to A3. But complex 5 where ATP is synthesized. And as I told you that ATP synthesis has two components, F0 and F1. F0 is a transport of proton or proton carrier and F1 is a catalytical molecule, and hence it helps in ATP synthesis, and that is what's shown in diagram clearly so as to understand what is ATP synthesis is here. This is ATP synthesis. With F1, F0 unit. ATP finally synthesized, proton release in matrix combined with oxygen for water. This is known as metabolic water. Why some of the organisms they aestivate and hibernate without any eating food? Why they live without water? During aestivation and hibernation, although they are on fasting, but respiration is going on. So with that respiration, water is produced. Therefore, they can live without water. Why kangaroo rat can live for 3 months, 4 months without drinking water? Because in its body, fats get oxidized and produces metabolic water. That metabolic water is sufficient to keep their water level in body. This is the physiology behind that some of the animals can drink water after 2 to 3 months. This is also very clear to you. I hope you enjoyed today's lecture. It's something combination of different knowledge, and therefore I have taken it as my privilege to explain you in a different way. I hope you got it, even though you practice it, and as a notes I have clearly mentioned everything is here. You can prepare this as a notes. You can note down these notes and write in examination for 5 marks. What is electron transport system? Explain it with schematic flowchart. What are 5 complex 1, 2, 3, 4, 5? Explain them. Or in NEET, any question can be asked. If this concept is clear, I hope you need not to worry. You can solve any question asked in NEET examination. So be prepared. I hope you did like today's lecture. Once again, I appeal you to share and subscribe our channel as well as press bell icon to receive notification as and when new videos are uploaded. Thank you. Enjoy.